Hello everyone, this is Ravindra Babu. Welcome back to my channel Silver 90s Biounit. By the last video, we successfully completed the salient features of a genetic code. Today, in this part of videos of molecular basis of inheritance, we are going to prove the salient features of a genetic code at a genetic basis through frame shifter mutations. Especially, we are going to prove the two salient features of a genetic code. Those are code is a triplet and code is continuous. Before we go into that, here a small request from my side, kindly watch all my videos made on this molecular basis of inheritance so that you can understand the concepts clearly and completely. And one more request is, please like and share the videos and subscribe my channel. Let's get into our today's topic, that is a genetic proof for that code is a triplet and code is a continuous. To make you understand this genetic proof for the salient features of a genetic code, let me give you a brief overview of mutations. We can define the mutations as the appearance of sudden heritable changes in the organisms of a population due to sudden changes in their genetic makeup. These mutations can be divided into majorly two categories. Those are chromosomal mutations and the second one is gene mutations. Chromosomal mutations means the changes in the genetic makeup occurs at the level of chromosomes. Gene mutations means the changes in the genetic makeup occurs at the level of a gene or below to the level of gene. Particularly, the changes occurs at the level of a basic structural unit of DNA, nucleotide. Hence the name gene mutations, and it is otherwise called point mutations. First, let us come to these chromosomal mutations. These chromosomal mutations are again divided into two. Those are chromosomal mutations due to change in number of chromosomes, and the other one is chromosomal mutations due to change in size and shape of chromosomes. These changing number of chromosomal mutations are again three types. Those are monoploidy, polyploidy and heteroploidy. This heteroploidy is otherwise called aneuploidy. Before we go into this monoploidy, polyploidy and heteroploidy, first we need to know what is a ploidy. Ploidy can be defined as any variation in the normal diploid set of chromosomes of an organism. Say suppose for example it is a diploid organism having 8 chromosomes, and these 8 chromosomes are in 4 pairs. Each pair is called a homologous chromosomal pair, which means the two chromosomes here are copies to each other. Hence the name homologous chromosomes, which means each chromosome in 2 sets. There are 4 chromosomes and each chromosome in 2 sets. To this what we call a diploid condition. If any variation occurs in this diploid set of chromosomes, then that is called ploidy. The ploidy is of how many types? 3 types here. First, monoploidy means it is the condition in the organism where the organism with a single set of chromosomes, and polyploidy means organism with more than 2 sets of chromosomes. If an additional set of chromosomes gets added to this diploid set of chromosomes, then it is called triploidy. This is what? A triploid condition. If 2 sets of chromosomes gets added to this diploid set, then it is said to be as tetraploidy. This is what about monoploidy and polyploidy. And the third one is heteroploidy. This heteroploidy is otherwise called aneuploidy. This heteroploidy can be defined as addition or deletion of one or more chromosomes to the complete diploid set of organism. In the case of monoploidy, we have seen a deletion of one complete set of chromosomes from a diploid set of chromosomes. This is what monoploidy. When we call it as the triploid condition, if addition of two sets of chromosomes to the complete diploid set of chromosomes of an organism, when we call it as tetraploid condition. But when we come to heteroploidy, it is not about the addition or deletion of a complete sets of chromosomes to the normal diploid sets of chromosomes of an organism, but it is the addition or deletion of chromosomes to the complete diploid set of chromosomes of an organism. Here the heteroploidy is of four types. These are trisomy, tetrasomy, monosomy and nullisomy. Trisomy is nothing but addition of one chromosome to the complete diploid set of chromosomes of an organism, which can be represented as 2n plus 1. This additional chromosome may be the copy of the fourth chromosome, or maybe the copy of a third chromosome, second chromosome or first chromosome, which means the number of possible trisomics in an organism is always equal to the haploid set of chromosomes of an organism. So this is the trisomy at a fourth chromosome. There is a possibility of a trisomy at a third chromosome or second chromosome. The multiple trisomics can be represented as 2n plus 1 plus 1 plus 1 plus 1 and so on. And if you come to tetrasomy, it is just addition of two chromosomes to the complete diploid set of chromosomes of an organism, which can be represented as 2n plus 2. This is a tetrasomy at a fourth chromosome, where two additional copies of chromosomes gets added to the fourth chromosome of a diploid set of an organism. This is tetrasomy at a fourth chromosome. Likewise, there is a possibility of occurrence of tetrasomy at a third chromosome, second chromosome and first chromosomes. Here also the possible number of tetrasomics equal to the haploid set of chromosomes of an organism. Multiple tetrasomics can be represented as 2n plus 2 plus 2 plus 2 and so on. Then if you come to monosomy, it is simply the deletion of one chromosome from the diploid set of chromosomes of an organism, which can be represented as 2n minus 1. The number of monosomics are always equal to haploid number of chromosomes of an organism. So this is a diploid set of an organism. If one chromosome gets deleted from this completed diploid set, this is said to be as monosomy at a fourth chromosome. Likewise, there is a possibility of occurrence of monosomy at a third chromosome, second chromosome or first chromosome. Here also the number of monosomics equal to the number of haploid set of chromosomes of an organism. Multiple monosomics can be represented as 2n minus 1 minus 1 minus 1 and so on. And the last, nullisomy means a deletion of two chromosomes from a diploid set of chromosomes of an organism, where it can be represented as 2n minus 2. If a complete homologous chromosomal pair gets deleted from a diploid set of chromosomes, then it is said to be as nullisomy of fourth chromosome, nothing but nullifying of a complete homologous chromosomal pair from a diploid set of chromosomes. Here also the number of nullisomics are always equal to the haploid number of chromosomes of an organism. Multiple nullisomics can be represented as 2n minus 2 minus 2 minus 2 like that. And now let us move on to the chromosomal mutations due to change in size and shape of chromosome. This type of mutations are also three. Those are deletions, duplications and translocations. Let us discuss one by one. Say deletion means the deletion of a segment of chromosome. Say suppose for example this is a chromosome having the sequence of genes A, B, C, D, E like this. If it involves in a deletion, say suppose for example if the segment of this chromosome having A, B genes gets deleted, after deletion the chromosome would be like this, having only C, D, E genes. This is what we call a deletion, where a segment of a chromosome gets deleted. And duplications, which means duplication of a segment of chromosome. Here this is the A, B, C, D, E, the sequence of genes on this chromosome. If it involves in duplication, say suppose for example A, B genes of this chromosome if gets duplicated, then what happens? The chromosome would be formed like this, having the duplicated copies of A, B genes. This is what we call duplication. And third one is translocation, which means the rearrangement or transfer of a chromosomal segment from one chromosome to another chromosome. So these are the two homologous chromosomal pairs in an organism. This is a one homologous pair, pair 1, and this is the homologous pair 2. If translocations occurs, say this segment of this chromosome of this homologous chromosomal pair may get added to the chromosome of this homologous chromosomal pair. What we call this? Translocation. Transfer of a chromosomal segment of one homologous chromosomal pair to another chromosome of another homologous chromosomal pair. This may happen reciprocally. Exchange of chromosomal segment between non-homologous chromosomal pairs like this. This segment of chromosome may get added to this homologous chromosome, and the segment of this chromosome may get added to the chromosome of this homologous chromosomal pair. What is happening here? Reciprocal exchange of chromosomal segments between non-homologous chromosomes, to which what we call translocation. Here there is a slight difference between translocation and crossing over. In crossing over, what happens? The exchange of chromosomal segments within the homologous chromosomal pair. But in the case of translocation, the exchange of chromosomal segments occur between non-homologous chromosomes. That is the basic difference between these translocations and normal crossing over, which occurs during S phase of interphase during cell cycle. This is about the chromosomal mutations due to change in chromosomal number and due to change in size and shape of chromosomes. Now let us move on to the next type of mutations. Those are gene mutations, which means the mutations occurs at or below to the level of genes. Especially the change in sequence of nucleotides of a gene is called a gene mutation. Since a small segment of DNA involves in these mutations, these type of mutations are otherwise called point mutations. Point mutations. Gene mutations and point mutations are same. These gene mutations or point mutations can be categorized into two. Those are substitutions and frame shifter mutations. First let us start with substitution mutations, which can be again divided into two types. One is transitions and another one is transversions. First let us start with the transitions, where the substitution of a purine by another purine and vice versa. Likewise, substitution of a pyrimidine by another pyrimidine and vice versa. Here the guanine may be substituted by adenine, adenine may be substituted by guanine, cytosine may be substituted by thymine, and thymine may be substituted by cytosine. When we come to transversions here, so this is the AT base pair. Here the adenine purine may get substituted by pyrimidine cytosine. This AT base pair would become CT base pair. Here the pyrimidine thymine may get substituted by a purine guanine, so that this AT pair would become a CG or GC base pair. This is what happens during transversions. The best example for transversions is sickle cell anemia. Let us discuss that sickle cell anemia. The sickle cell anemia is a genetic disease which is caused due to substitution mutations, particularly by transversions. Generally, the hemoglobin of a human being is composed of 4 types of polypeptide chains. Among those 4 types, 2 are alpha globin chains and 2 are beta globin chains. This second beta globin chain is composed of 147 amino acids. Among these 147 amino acids, the 6th amino acid is a glutamic acid. When substitution mutations or transversions takes place, this 6th amino acid glutamine may replaced by another amino acid valine, which lead to the causing of the genetic disease sickle cell anemia in human beings. In this disease, the RBC cells are normally in sickle shaped instead of biconcave disc shaped structures. As these are abnormal in shape, they cannot move freely in the blood vessels. Here the question is how this sickle cell anemia is a best example for transversion mutations. Let us come to that. So this is a gene or DNA which is coding for the second beta globin chain of hemoglobin. Here this is the sequence of nucleotides on the DNA molecule. When this DNA molecule involves in transcription, it produces a mRNA molecule having the sequence of triplet codons like this. This is the 6th triplet codon on the DNA and this is the 6th triplet codon on mRNA, excepting this first initiation codon. Whenever this mRNA molecule involves in a translation process, it would produce a polypeptide chain having the sequence of amino acids like this. This is the glutamic acid, which is the 6th amino acid of this polypeptide chain. Whenever a substitution mutation or transversion takes place at this 6th triplet codon of DNA, what is the transversion? The transversion means the substitution of a pyrimidine by another purine or substitution of a purine by another pyrimidine. Here the pyrimidine thymine gets substituted by a purine adenine. Then what happens? CTC will become CAC. If this DNA molecule involves in transcription, it would produce an mRNA molecule having the 6th triplet codon as GUG. Whenever this mRNA molecule underwent mutation involves in translation, it would produce the amino acid valine at this 6th position instead of glutamic acid, because the mutated triplet codon on the mRNA is not a GAG, it is GUG. This led to the cause of a genetic disease, sickle cell anemia. Now let us move on to the second type of gene mutations. Those are frame shifter mutations. The frame shift mutations can be defined as insertion or deletion of one or more nucleotides in DNA results into shift in reading frame of triplet codons on mRNA molecule, thereby shift in reading frame in the sequence of amino acids of a polypeptide chain. Let us see that mechanism of frame shifter mutations. So this is the mRNA molecule having the sequence of triplet codons like this. If this mRNA molecule without frame shift mutations, if it undergoes a translation, it would produce a polypeptide chain having the sequence of amino acids like this: methionine, valine, isoleucine, phenylalanine and alanine. This mRNA molecule, if it undergoes a deletion of frame shift mutation, then what happens? For example, here I am taking this adenine nucleotide of this third triplet codon on mRNA. If this adenine, it gets deleted, The third triplet codon would be UCU, as A gets deleted here. This UC with the first nucleotide of the adjacent triplet codon would form a triplet codon, hence UCU. And next the two nucleotides of this triplet codon UUU gets combined with the first nucleotide of adjacent triplet codon G to form a triplet codon UUG. Like that, the two nucleotides CA of this triplet codon by combining with the first nucleotide of adjacent triplet codon would form another triplet codon. Nothing but what happened here, from the point of deletion you can find a shift in reading frame of triplet codons on the mRNA molecule. Such type of mRNA molecule, if it undergo translation, it would produce a polypeptide chain having the sequence of amino acids like this. The sequence of amino acids are methionine, valine, serine and leucine. Here also you can find a shift in reading frame of sequence of amino acids from the point of deletion of a nucleotide on the DNA or RNA molecule. Here the deletion has taken place, so that from the point of this deletion, here you can find shift in reading frame in the sequence of amino acids in this polypeptide chain. Likewise, the insertional frame shifter mutations. Here additional nucleotide gets added to this mRNA molecule due to insertion. Say suppose for example here the nucleotide of guanosine gets added before this adenosine nucleotide. Then what happens? The mRNA molecule would produce having the sequence of triplet codons like this AUG, GUA, but here you will get a GAU. What is happening here? The G, A and U will form a triplet codon like this. The remaining C of this triplet codon with the next two nucleotides of the adjacent triplet codon would form CUU triplet codon. Here the remaining U nucleotide, which by combining the two nucleotides of adjacent triplet codon would form UGC triplet codon like this. Nothing but here also what happened? A shift in reading frame from the point of addition of a nucleotide. Up to here the sequence of triplet codons is same AUG, GUA, AUG, GUA. But from this point of insertion you will find a shift in reading frame in the triplet codons of mRNA molecule. Such a mRNA molecule, if it involves in the translation process, it would produce a polypeptide chain having the sequence of amino acids like this: methionine, valine, aspartic acid, leucine and cysteine. In the normal polypeptide chain, the sequence of amino acids is methionine, valine, isoleucine, phenylalanine and alanine. From this point of insertion of a nucleotide, you will find the shift in reading frame of the sequence of amino acids. This is what we call the frame shifter mutations. This is all for today's video about mutations. Actually, I wanted to continue the genetic proof for the code as a triplet and continuous. In the next video, we will complete this genetic proof for the code as triplet and continuous. Thanks for watching. Like and share the videos and subscribe the channel. Vidya Amrutam Ashnute.